For the past several months, our group has been researching an outbreak of an unknown disease along the Gulf Coast. These are our findings of azzius disease. Azzius disease is vectored by Anopheles braidlii. Anopheles braidlii is within the Anopheles genus. The Anopheles genus contains about 450 individual species, and many of these species transmit disease to humans. Anopheles genus prefers to feed on humans, although they do feed on other animals. Anopheles mosquitoes can kill over 400,000 people a year worldwide due to their vectored diseases. Anopheles braidlii species is known to vector several diseases, including malaria, eastern equine encephalitis, and West Nile virus, and our disease, azzius disease. This map shows the distribution of the Anopheles genus worldwide. The Anopheles genus are found all over the world and not just in tropical climates. Anopheles are found worldwide on every continent except for Antarctica, and because the Anopheles are found worldwide, there is a constant threat of infestation of Anopheles mosquito-borne diseases. This diagram shows the life cycle of an Anopheles mosquito. So, the Anopheles braidlii mosquito has 4 life stages, including an egg, larva, pupa, and an adult. To begin the life cycle, a female Anopheles mosquito will lay each of her eggs individually on the surface of the water. Sources of water can include streams and lakes. These eggs that she lays are elongate and oval-shaped. When they're laid, they are clear white in color and gradually darken over time, and the eggs can hatch anywhere from 2 to 3 days to weeks at a time. Although most aquatic larvae breathe through a siphon, the siphon is absent in the Anopheles larvae. Instead, they breathe through spiracular openings, and because of these spiracular openings, Anopheles larvae position themselves parallel to the surface of the water. Larvae go through 4 larval instars for development before becoming pupa, with each instar taking only a few days. The pupal stage can last up to 2 weeks before they emerge as adults. Adult males feed on nectar and sugar. Females will feed on nectar but require blood meals to produce eggs. And typically, adult mosquitoes will live for 1 to 2 weeks. So this map shows the habitat of Anopheles braidlii. Anopheles braidlii is commonly found along the Gulf Coast and the East Coast of the United States. This is shown here on our map, and it is in line with the locations of each of our azzius disease patients.
Our disease agent is a previously undiscovered bacteria known as Proteus azzius. Proteus azzius is a bacillus-shaped gram-negative bacterium. Proteus azzius has a genome size of 4.49 megabases, which allowed us to identify it as a bacterium. The genus Proteus is in the family Enterobacteria. These bacterium are characterized by their production of acetyl-methyl-carbinol during glucose metabolism, and their ability to reduce sulfur compounds to sulfides. The species azzius was previously undiscovered. This species produces acid during the fermentation of glycerol and is able to produce tryptophanase. The bacterium Proteus azzius causes azzius disease, which has resulted in several deaths.
10 patients were the focus of our investigation. These patients were primarily found on the Gulf Coast of the United States, including Florida, Louisiana, Texas, Mississippi, and Georgia. The disease was brought into the United States by our patient 0, Jeff Blumkin. Blumkin was introduced to the disease during travels abroad in Asia, where he came in contact with an infected rat species. He brought our unknown disease, Proteus azzius, into the United States upon his return. The bacterial disease was spread among the 10 patients across the southern coast. These patients all shared severe major symptoms including meningitis, fever, headache, body aches, exhaustion, dehydration, and eventually death. Let us now take a moment to remember these brave Americans.
We identified our reservoir as small rodents, including field mice, rabbits, and rats. These animals are commonly pests and never venture far from their homes. They're active all year long and are able to avoid predators by staying close to their burrows, and they store food in order to survive long winters. These rodents are found across the world, particularly in North America, Europe, and Asia. Specifically, our disease was brought to the United States on rats from China. Because of their versatileness, they are able to survive in a variety of environments, though they prefer areas where food is abundant and they can conceal themselves. They are common prey of the Anopheles braidlii and other female mosquitoes because the mosquitoes are able to sense the warm blood of the rodents.
We will now move on to our final comprehensive control plan.
Initially, to control our vector, we chose to spray mosquito pesticides from trucks at night. While this spraying was relatively effective against adult mosquitoes, it had little to no effect on mosquito larvae. Additionally, very few local residents picked up pesticide machines to spray at their own homes, and there were many concerns about negative health consequences, particularly in children. Environmental groups were concerned that this pesticide spraying would kill not only Anopheles braidlii, but many other mosquito and insect species in general, which would have detrimental effects for insectivores. Activist groups also had a lot of trouble with our pesticide spraying, and their protests received national news coverage. Further, the governors of both Florida and Texas made very public threats against our control plan because they were upset that the federal government had overstepped its bounds by interfering in local matters.
Wetland habitats and dense grassy areas can favor mosquito production. These areas can harbor adult mosquitoes and provide areas for egg production. Vegetation provides shade, cover, and protection for all mosquito species. To combat this, a government-led campaign to tame overgrown vegetation could greatly reduce the Anopheles braidlii species habitat. This campaign could include managing grasses on sides of roads and highways, maintaining lake areas and shores, and providing routine lawn maintenance to local parks. In addition to these methods, local governments should include their residents to regularly mow yards and trim weeds. This will reduce mosquito resting areas and habitats around local homes. A combination of these methods promoted by the government over the course of a year could drastically control adult mosquito numbers without invading the habitats of local animal species.
We will be using mosquito fish to control larval Anopheles mosquitoes. Mosquitoes lay their eggs in water, and after the eggs hatch, they become mosquito larvae. The mosquito fish, Gambusia affinis, is a species of freshwater fish that is used frequently by mosquito control organizations. Mosquito fish are efficient for mosquito larvae control due to their ability to eat hundreds of mosquito larvae a day. Mosquito fish have been successfully used by New Jersey's Department of Environmental Protection to kill larval mosquitoes that will potentially carry pathogens in the future. We will thus be following their published protocol on mosquito fish implementation, where solutions to potential problems are addressed. We will be following a year-long timeline, hopefully breaking the vector to reservoir link in the disease chain.
Now we move on to aquatic habitat source reduction, which is very important because it is a relatively simple way of controlling the mosquito population in an area. By reducing the number of places that mosquitoes have to mate and oviposit, we effectively make it harder for their numbers to grow and therefore harder for diseases to spread. This type of source reduction is also fairly simple. Removing things such as tires, pools, empty flower pots, and many others can have a substantial impact in reducing the mosquito population in an area. The team will partner with the local governments of impacted areas as well as with local governments of the surrounding areas in order to start an education campaign to help the communities understand the hazard and how easily they can be prevented. Proper implementation of this method would likely ensure that the habitats in which mosquitoes tend to live and develop in would be eliminated, and subsequently transmission of the diseases in the affected areas. After doing some research, we discovered that plant extracts may be used as a larvicide to control mosquitoes in their early stages. Specifically, the crude petroleum ether extract of Croton sparciflorus was found effective against Anopheles. Croton sparciflorus is a common weed in South India and can be imported into the United States. This method of vector control is favorable because the larvicide is applied directly to bodies of water, unlike traditional pesticides. Any body of water can be treated and the larvicide will specifically target mosquito larvae. This method can be utilized in areas that condemn the use of synthetic chemical larvicide. This approach will require the effort of the team, local communities, and local governments in order to actively perform the larviciding treatment, as well as to promote the continued use of the process.
While the mosquito spraying was highly effective at breaking the host vector link, we abandoned this control approach due to the large public backlash. The vegetation control breaks the link between host and vector, removing the adult mosquito habitat and thus preventing them from reaching local residents and communities. Additionally, maintaining these grassy areas will prevent Anopheles bradleyi from reaching the small rodents that may carry azzius disease, thus breaking the vector reservoir link in the chain. Mosquito fish control breaks the vector reservoir link. By attacking mosquitoes at their larval stage, we are able to prevent the possibility for mosquitoes to mature and come in contact with the reservoir. Ultimately, this eliminates infectious mosquitoes in the environment and contributes to an effective disease control protocol. Aquatic habitat reduction is very important to this control plan. The process helps to break both the vector reservoir link and the vector host link. This is due to the fact that since all breeding and mating sites are removed, there is no chance that larval or pupal mosquitoes will survive to become adults and transmit the disease. This same principle carries over to larvicide, which is one of the most effective vector reservoir links because it kills all larvae before they are able to pupate, thus removing them from the disease cycle.
Thank you all for listening to our disease control presentation. Are there any questions?
I had a question about your disease prevention plan.
Okay, great. If there are no further questions, then this is the end of our presentation. Thank you.